let's discuss those tRNAs a little bit more. What is the structure? What is the function specifically of transfer RNA? Each tRNA will escort in one specific amino acid. The shape of the tRNA will allow for a bend in the RNA strand that will expose 3 RNA nucleotides. Those 3 RNA nucleotides are known as the anticodon. The messenger RNA has the codon, the triplet, remember. The tRNA has the complement to that, which is referred to as the anticodon. If the mRNA codon says CCC, the anticodon is GGG. Overall, the tRNA itself, the RNA strand, is relatively small. Okay, it's only about 80 nucleotides in length. It folds in on itself, even though the RNA strand that is what we would think of as the tRNA is only 80 nucleotides in length. It will fold in on itself. It will complementary base pair with itself, and it begins forming these secondary, tertiary structures. These secondary, tertiary structures that gives it a three-dimensional shape. Remember, shape dictates function. When we draw it out, okay, we humans are simple-minded. So we draw out this thing we see over here. This thing we see over here, this clover leaf looking thing, you see down here, the anticodon. Here, the artist has drawn it, AAG. So that means the codon in the messenger RNA will be UUC. Up here, this 3' end, this is where the amino acid will attach. We may draw it this way, but the thing is, we are very simple-minded. Simplistic drawing like this are not reality. This is reality. This is what the tRNA looks like. This is what the artist draws because it's so much easier to draw that. What happens? This curves over. This arm comes this way. That arm comes that way, like it's hugging itself. There you go. Here's this L shape. The specificity. tRNA that has the AAG anticodon will have one specific amino acid that it will escort in. Because remember the codon table? And no, I can't remember what UUC is off the top of my head. That codon table, degenerate, but there's no imprecision to it. So we do know that for every codon, there is a complementary anticodon, and that anticodon, that tRNA, will bind to one and only one type of amino acid. How that amino acid gets bound to the 3' end is done by aminoacyl tRNA synthetase. Aminoacyl tRNA synthetase will interact with tRNAs until it gets the right tRNA with the right anticodon, and then it will bond the amino acid to it. One of the things we found, if you go and you look back at that codon table, if you look at the pattern, those amino acids that have multiple codons that will signal for it, will call for it, you'll notice that the first nucleotide in the codon is always the same. The second one generally is always the same. There may be some that have a second that could be changed, but the third is where variability may be allowed. So GGC will call for the same amino acid as GGG. They refer to this as wobble. Okay. Wobble allows for some tRNAs to escort in, or multiple tRNAs, one amino acid. It also allows for the potential of a mutation. What should be GGG, a point mutation, means that the change to GGC will have no effect. What this also means is that you do not need 64, or 61, sorry, 3 codons code for nothing. You do not need 61 different tRNA synthetases. No. What you can have is like 16 of them, I think it is. 16 of them that will actually interact with the first nucleotide in the anticodon specifically, the second one with a little less specificity, and the third one can vary depending upon which the amino acid that specific tRNA synthetase is escorting. You notice here in this example, tyrosine tRNA synthetase. Well, this tRNA synthetase only interacts with the amino acid tyrosine. Does not interact with serine, isoleucine, leucine, any of the others. It will bind up the free amino acids in one of its sites, and then it'll wait for an uncharged tRNA, one that has the specific complementary anticodon AUA. AUA will fit into the active site, a little bit of ATP, and then a new bond is formed. That tRNA is now charged because tyrosine is covalently bonded to it.